Let's say you're on the ward. You notice a distinct pattern in your patients, something that happens repeatedly but doesn't quite fit the textbook. You step into the corridor, pull out a notepad, and write it down. You think you have a great research idea. Later, you log into PubMed to map out your study, but within 10 minutes, you hit a wall. A paper from 5 years ago already studied this exact clinical pattern. Your idea is already out there. This is a common point of frustration for early career researchers. Most broad clinical ideas have been explored in some form, but finding existing literature does not mean your initial observation is useless. The issue lies in understanding what academic journals actually value. They rarely publish broad ideas. They publish specific gaps. A gap is a strict boundary line. It answers exactly what remains unknown, where the evidence conflicts, or what specific protocol has not yet been applied to your precise patient population or hospital setting. If you submit a manuscript based solely on a sharp clinical observation, you invite an automatic desk rejection for lacking novelty. To survive peer review, you have to find and name the gap. Finding that gap starts with step 1, observe. You do not need a complex, perfectly formatted research question on day 1. You only need a single, simple sentence derived directly from what you see in daily practice. Let's use a running case study. Here is our initial observation. Diabetic inpatients on my service seem to fall more often. Once you have that seed, you move to step 2, broaden. We take this single sentence and deliberately explode it into multiple variables before choosing a direction. We look at the immediate clinical factors. Is this increased fall risk related to hypoglycemia? Is it driven by severe neuropathy? Or perhaps it correlates with the use of sedative medications? We also look at systemic and environmental factors. Could it be polypharmacy, poor mobility, visual impairment, or maybe a correlation with overall hospital length of stay? The rule right now is simple. Do not pick a favorite angle yet. You have to let the existing literature dictate which path is actually worth taking. By forcing yourself to map out these distinct variables, you avoid designing an entire study around a question that another research team conclusively answered a decade ago. Step 3 is the rapid literature scan. This involves scanning roughly 40 papers in a single afternoon, rather than reading each one cover to cover. To execute this, we use a specific stack of AI tools to accelerate the process. This visual maps out a workflow combining PubMed with several specialized engines. First, use Perplexity. It provides a broad, sourced overview of your clinical topic, pointing out the major concepts and recent discussions. Next, search Semantic Scholar. This lets you quickly digest short, accurate summaries of highly cited studies without getting bogged down in the full text. From there, plug your specific variables into Consensus. It aggregates the data to tell you if the majority of existing studies broadly agree or disagree on a given point. For the detailed data, use Elicit. It extracts specific clinical claims, patient demographics, and outcomes from multiple papers into a structured matrix you can read at a glance. Finally, use ResearchRabbit to visually map out citation networks. This ensures you discover the older foundational papers that everyone else is citing. The point of this sprint is purely reconnaissance. You are checking to see if there is enough uncharted territory within your broadened angles to justify investing the time into a full project. Once you complete a scan, you move to step 4, sort. Your job is to filter the evidence you gathered into 3 distinct conceptual categories. This diagram shows our 3 buckets: established, conflicting, and missing. The first bucket is established. This contains facts the literature universally agrees on, like the basic premise that diabetes increases fall risk in older adults. If you build a research question based on what is in this bucket, you will hit a dead end. The only exception is if you apply a radically different methodology or study an entirely unexamined population. The second bucket is conflicting. This captures active debates. For instance, you will find that studies disagree on whether glucose variability independently predicts falls when adjusting for frailty. Conflicting evidence is highly valuable. It gives you a clear target. Your study can provide the exact data needed to settle an ongoing academic argument. The third bucket is missing. This holds the variables that researchers simply haven't tested yet. In our diabetes example, maybe nobody has examined inpatient glucose variability specifically within Middle Eastern hospital settings. This missing space is typically your strongest possible location for a highly publishable, defensible research gap. The defensible gap lives in those second and third buckets. This approach changes the focus of the literature review from exhaustive reading to objective-driven sorting. With your evidence sorted, you start step 5. Name the gap. You have to distill your chosen target into one clear, declarative sentence. Look at this comparison. A weak gap statement says, there is not enough research on diabetes and falls. The strong gap statement says, although diabetes is associated with fall risk, limited evidence examines whether inpatient glucose variability predicts falls among hospitalized adults with diabetes in Middle Eastern healthcare settings. Notice the structure. It acknowledges the established area, pinpoints the specific missing variable, and locks down the exact target population and clinical setting. Finally, step 6, refine for feasibility. A theoretically perfect gap will fail if you don't have the time, the data access, or the ethical approvals to actually execute the study. We have to narrow our diabetes observation further. We constrain the data to patients admitted to medical wards between 2022 and 2025. And instead of the broad concept of glucose variability, we isolate the study strictly to documented hypoglycemia. Our refined gap is now highly specific. Among hospitalized adults with type 2 diabetes admitted to medical wards between 2022 and 2025, is documented hypoglycemia during admission associated with inpatient falls. A tightly constrained project that you can realistically complete and publish is universally superior to a massive, ambitious idea that stalls in the ethics committee. Before you formalize your protocol, there is a critical rule regarding AI in scientific research. Artificial intelligence accelerates the discovery process and organizes the data, but it does not validate the underlying science. You must always open the original paper. You verify the DOI, you read the abstract, and you confirm that the authors actually made the claim the AI says they made. Never cite an AI-generated summary in a manuscript. You must read and cite the primary source. Follow this pipeline to engineer your idea. Observe the clinical problem. Scan the literature rapidly. Sort the evidence into buckets. Name your specific gap. And refine the boundaries until it is fully feasible. If you complete these steps, you now possess a gap-backed, publishable idea, but you do not yet have a final operational research question. Transforming that gap into a precise protocol requires structuring it through the P-code and finer frameworks. We will cover that exact process in the next lecture. To immediately begin engineering your own defensible research gaps, head to BayyinahResearch.com to download the AI prompt library and the literature review workflow.